Hello again and welcome to part 3 of my beginner's guide to colour genetics in Budgerigars. And in part 3 we're going to take a look at the codominant varieties of Budgerigar and how we can predict the outcomes of those Budgerigars. If you haven't watched the first two parts of this guide, then please do go back and have a look at them. Also, don't forget, it would be really welcome to hit the like button and to subscribe to the channel. Any comments, please do leave those. So let's now have a look at codominance. Well, before we look at the codominance variety and those predictions, let's have a quick recap on what you should know before we do that. So in the last video we looked at how the interaction between dominant varieties and recessive varieties, so dominant allele and recessive allele. And from those you will remember that whilst there were 3 genotypes, there were only 2 phenotypes. So with a dominant recessive variety, then there are 3 genotypes and only 2 phenotypes. You'll also remember that we used a number of letters and varieties to represent the mutated and wild type genes, and we also represented the dominant type in uppercase or capital and the recessive in lowercase. Some of those also included a number, you may remember. And finally, and probably most importantly, we learnt how we would use the Punnett Square. And I say most importantly because once again in this video we're going to be using the Punnett Square to look at how we predict the dominant, the codominant varieties. So you'll need to have an understanding of, or the basic understanding of, how to use the Punnett Square in order to progress there. And like I say, if you are unsure about the Punnett Square, want to have a quick look back on that, then do go back and review part 2 of this genetic guide, and I will put a quick link to part 2 across the top of the screen now. So then let's have a look at codominant varieties. So the first thing we need to think about when we're talking about the codominance is something that you'll hear quite often people referring to, and that's single and double factor. You will remember from, I think it was from part 1 of this guide, that every Budgerigar will contain 2 genes, or 2 of each gene, so 2 allele for each of the genes, and those genes are contained on chromosomes. For this video we don't need to worry too much about the chromosome, but just the understanding that each Budgerigar contains 2 of each of the genes. Before those early geneticists understood that there were genes, they referred to these as factors because they understood that there were two things interacting to produce a particular outcome in the offspring, and they referred to those as factors, and we still use those now when we're talking about genetics. We still talk about the various factors, and they refer to those factors, or a animal or a plant, as being either of a single factor or of a double factor. And all that really means is a single factor contains one of the mutated genes, so one allele of a mutated gene, and a double factor contains 2 mutations of a particular allele. So there's nothing new, nothing special about this in terms of how we understand it. So if you hear someone talking about a single factor, they merely mean that it contains one of the mutated allele, and if they're talking about a double factor bird, then it contains 2 of the mutated allele. In terms of the codominance, quite often when they talk about single and double factor, they're also referring to the phenotype of that particular gene, and I'll come on to that. I'll talk a little bit more about that in a bit. But in effect, even the dominant and recessive varieties come in single and double factor forms. It's just that the single and double factor variety in a dominant variety of Budgerigar will both look the same, because as we know, in dominant recessive, then if the bird contains one of the dominant allele, it will always look like it. So a single factor and a double factor dominant recessive variety will always look like the dominant version of that particular variety. I hope that makes sense. So that's all we mean when we're talking about single and double factor. It's the number of mutated allele that particular bird has, and it particularly we particularly talk about it when we are talking about codominant varieties. So what I thought I'd do is just quickly introduce a couple of the more common codominant varieties, so we get a bit of an understanding of what we're talking about. So the first one I'll briefly mention is the dark factor. There again, I'm using the word factor there. So, and we know the dark factor comes in, or the dark factor does come in a single and double factor, and that in effect impacts on how dark the bird will be. So if it contains no double factors, then it will, or no dark factors, then it will be the normal light green or sky blue. And then if it contains a single factor or single dark factor mutated allele, then it will look like either the cobalt or dark green, and the double will be mauve or olive. So there you can begin to see how the various interactions work when we're talking about the dark factor. But it's probably more noticeable in a variety like the Spangle variety, where there is some real clear differences between a single factor Spangle and a dark factor Spangle. So single factor Spangle and a double factor Spangle. And we'll have a look at that by going over to the Punnett Square, and we'll look at the outcome of, or predicting the outcome of breeding two single factor Spangles together, and that will give us the best indication of how codominant varieties work. So here we are back at the very familiar Punnett Square that you will remember from the last video. Just a quick reminder of how this works. Across the top up here we've got the what we're pairing together, in this case both single factor Spangles and a single factor Spangle. We're not worried at this stage about the colour. We'll come on to that in a future video about how we work out the colour as well as the variety. And so we've got a single factor Spangle across the top here, normally the cockbird, and the single factor Spangle again down the side, normally the henbird. And these are the various allele for the Spangle gene, obviously two for each of the birds. Where we've got a codominant variety, then it's considered to be dominant, so the uppercase S shows the mutated Spangle gene, and the lowercase s would show the wild type non-Spangle gene. So how do we work out what the possible outcomes from this pairing is going to be? Well, you remember from the last video, what we do is we bring down the top allele from this square and place it in here. So that's an uppercase mutated allele, so the uppercase S, and we bring across the allele from the hen bird as well, so that goes across to there, and that will give us another uppercase S. And then with this square here we will produce the lowercase non-mutated lowercase s, and of course an uppercase mutated allele there from the hen over here. I'll just fill the rest of these out. So the uppercase mutated allele and the lowercase allele, and then here we've got lowercase non-mutated allele and the lowercase non-mutated allele there. So in this case we've got all three of the possible genotypes. So up here we've got two allele for the mutated Spangle So we said there's 2, so that's double, so that's a double factor Spangle. Over here we've got 1 mutated allele, and over here we've got 1 mutated allele, and so that will give us a single factor Spangle, and here there are none, so that would give us a normal, so a non-Spangle bird. So from 2 single factor Spangle pairings, so from a pairing 2 single factor Spangles together, we would in effect get 50% of the birds. So we can see from this square here and this square here, 50% of the birds will be single factor Spangles, 25% of the offspring would be double factor Spangles, and 25% would be a normal bird. So that's averaging it out. So we would get 50% single factor Spangles, 25% double factor Spangles, and 25% normals. So there we go, that's how you work out the codominant varieties. They all would work out exactly the same. If you want some practice, try redoing this Punnett Square, but using it with dark factor in there, and see moving it around so you can see what you would get if you put in a say a dark factor with a normal, and seeing what the possible outcomes of that would be. Or indeed, why not do one here with a double factor Spangle and a single factor Spangle and see what that would produce. What I will do is I will do that now. So I'll do a double factor Spangle and a single factor Spangle. You can then work out what the outcome is going to be and then check it with mine. So there we go then, there's the double factor and cross single factor Spangle Punnett Square with it all partially filled in. See if you can complete the square and work out the percentage outcome for this particular pairing. What I'll do now is if you pause the video and work that out, and then we'll come back and I will have completed it. And so there is the completed Punnett Square for this, and as you would see, you will get 50% of the birds will be double factor Spangles and 50% of the birds would be single factor Spangles, so a 50-50 split there. Notice you will get no normals from this particular pairing. And there we are, that's the codominant variety Punnett Square. So there we go, that's how we predict the codominant varieties. Fairly straightforward. I suppose the only thing you really need to remember is that with any codominant variety there will always be an in-between stage. So a bird that looks different from the wild type but also different from the double factor. So in effect, unlike the dominant and recessive birds where we had 3 genotypes but only 2 phenotypes, there is a phenotype for each of the genotypes. So 3 genotypes and 3 phenotypes. Well, I hope you enjoyed part 3. As always, if you want to go over this in a bit more depth, then do pop across to the Shed Talk Facebook page, where my Beginner's Guide to Genetics PDF book is available. It should be right at the very top of the Facebook page. Free of charge to download, and takes you through all of the videos of this series in a little bit more detail, and talks you through some of those nuances around the genetics, like I say, in a bit more detail. In the next video, we're going to take this a stage further. As we all know, Budgerigars don't just come in a single variety. So, you know, let's think about the Spangle in particular. You're going to have both blue series Spangle and green series Spangle. So what do you get, or how do you use a Punnett Square to predict when you've got more than one allele or more than one mutation to consider? Say you've got the blue mutation and the Spangle mutation to consider. So that's what we're going to focus on in the next video, but until then, if you enjoy the videos, then please do hit the like button and of course do subscribe.